Welcome, Junior Tuckies. My name is Mrs. Ramla. This video I have prepared for the grade 12s to help with their grade 12 final exam preparation. In this video, I will look at the Feb-March 2013 paper 1. Right, 1.2. Give the correct biological term for each of the following descriptions. Write down only the term next to the question number in the answer book. 1.1. The linking of the present-day distribution of organisms with the past movement of continental plates. That we know is biogeography. 1.2. The elimination of a species from the earth, that we know as extinction. 1.3. The point where the exchange of genetic material occurs during crossing over. They're saying here the point where the exchange takes place, that's the chiasma. 1.4. The stage in the process of the formation of a protein determined by the codons in the mRNA. What stage of protein synthesis is this? Right, that is obviously translation. 1.5. The type of inheritance which has a range of intermediate phenotypes for a particular trait. Of course, we know that as polygenic inheritance. 1.6. An inherited disorder in which blood fails to clot properly. We know that's hemophilia. Look at the spelling of hemophilia. 1.7. The position of a gene in a chromosome. That's a locus. Now, you might be quite confused and wondering why is there evolution in this paper. In 2013, in those days, the sections were still a bit different. But we will carry on because you still tested on evolution, so this paper will still be of benefit to you. Right, 1.3. Indicate whether each of the statements in column 1 applies to A only, B only, both A and B or none of the items in column 2. What must you write down? A only, B only, both A and B or none, next to the question number in the answer booklet. 131. Who discovered the double helical structure of the DNA? Is it Francis Crick, James Watson? Well, you should know that it is both of them, James Watson and Francis Crick, that discovered the structure of the DNA. So your answer, can you see? Both A and B. 132. A process which can detect mutant genes in an individual. Is it genetic counseling? No. From its name, it's counseling. Is it genetic testing? Yes. And genetic testing is used to determine or detect mutant genes in an individual. So your answer is obviously B only. 133. The breeding of organisms over many generations in order to achieve a desirable phenotype. Is it cloning? No, cloning is not breeding of organisms over many generations. Yes, we know it is artificial selection. So your answer is B only. 134. What introduces variation within a species? We know it's random mating and we know it's mutation as well. So here the answer is both A and B. 135. An advantage of polyploidy in agriculture. Polyploidy is when a cell has more than 2 sets of chromosomes. Could be triploid or tetraploid. What does this do for a plant? Obviously, it's going to cause the increase in the size of fruit or increase in the size of flowers. So your answer, of course, is both A and B. 136. A type of cell division that takes place in angiosperm plants. Of course, we know mitosis and meiosis takes place, so it's both A and B. 137. What influences the inheritance of blood groups? Again here, certain blood groups A and B is codominant and it's influenced by multiple alleles. So your answer is both A and B. And the last one, what are the bonds that hold amino acids together in a protein? We know hydrogen bonds is not the answer. We know the answer is peptide bonds only. So B only. Now in 2013, there should be 8 of these questions. But now, in the current papers, there's only 3 of them. Because in the past, the learners never used to write both A and B only. And the learners used to lose a lot of marks. So now, there's only 3 of these questions. Right, 1.4. The pedigree diagram below traces the inheritance of vestigial, which are reduced in size and normal wing characteristics in fruit flies. Study the diagram and answer the questions that follow. 141. State the dominant wing characteristic of flies using these crosses. Now, the clear blocks is vestigial female fruit flies or vestigial male fruit flies, and the shaded block and circle is normal fruit flies. meaning they have normal size wings. Now, state the dominant characteristic. Now, if you look at it, you can see that there's 1, 2, 3, 4, 5, 6, 7. 7 of these members in the family are shaded in. So that means they have normal wings. So, and there's only 3 that have vestigial. So of course, what's the dominant characteristic, wing characteristic? Normal wings. 142. Use the letters G, big G and small g, and write the genotypes of the following. A. Now, A, can you see? A has to be big G because it's normal and it's dominant. But can you see A has a child that's small g, and the only way A can have a child with the small g is if A is heterozygous. So A is big G, small g. Then of course, use the letters big G and small g and write on the genotype of J. J of course is the recessive allele, so it can only be small g. Okay. Now, it's a very nice question, this. You must take your time and you must work this out. Now remember, genotype means genetic makeup, so you use the letters. Okay. 143. If fruit fly C, which was big G, small d, was crossed with a male having vestigial wings, what will be the possible genotypes of the offspring? There's your answer here, but I included it how you work it out. So big G, small g, crossed with small g, and there's your gametes. There's your offspring. Okay, that wasn't such a bad question. Right, 2.1. The diagram below shows part of a DNA molecule in a nucleus just before cell division. 2.1. Identify parts 3. Now, can you see part 3 is this part here? It's the part that's shaped like this. Can you see it has 5 sides? That's the sugar. So 3, this is a DNA, so the sugar is a deoxyribose sugar. Right. 4. 4 is a little circle part that joins the phosphate, that joins the sugar to the nitrogenous base, and that we know is the phosphate group. Don't just say phosphate, say phosphate group. 212. Identify the nitrogenous base labeled 1. Here we are here. Now can you see it's 1, 2, 3, 4, 5. Now on this side, 1, 2, 3, 4, 5. So this is cytosine. What's the complementary base for cytosine? Guanine. Right. 1. That was 1. Then identify nitrogenous base 2. 1, 2, 3, 1, 2, 3. There's it there. So this is the base, which is the exact same one here. Also guanine. You just have to focus and look at the diagrams and how it's forming. 213. Explain why the diagram above represents replication and not transcription. Right. The structure that's formed, the strand that's formed, can you see everywhere? There's just thymine. There's no uracil. Okay. Both strands, can you see each strand on either side is being used as a template. So that's how we know it is DNA replication. Right. This is my favorite question. A rare form of rickets in humans is caused by sex-linked dominant allele, big R, which is carried on the X chromosome. An affected female whose father was unaffected married an unaffected male. Now, big R, this is now it's different. It's sex-linked and it's dominant. And small r is normal. So, they want you, it was an affected female. She's affected. And her father was unaffected. So there's a genotype here, X big R, X small r. How do I know she's small r? Because her father was unaffected. Now what does she marry? She marries an unaffected male. So look, X small r Y. Why am I only putting it on the X? Because it's an X-linked condition. It tells you that. I love this question. So there we are. We've determined the possible genotypes, phenotypes of the offspring. Now we must do a genetic cross. So there's the gametes. We wrote P1, we wrote affected female, marrying an unaffected male. We got the genotype. Then we write meiosis, gametes, fertilization. And then I prefer to use the Punnett square. And then you write your gametes out here. And then your offspring. And then your phenotype. She will have one affected daughter, one affected son, one unaffected daughter, one unaffected son. Right. So then they ask you the possible genotypes and phenotypes. So this is all they want you to write it down carefully. Okay, this is all for 6 marks, as you can see in the memo. 2.2.2, what is the percentage chance that they will have a child who is unaffected? Now you can see it's 1 out of 4. So 1 out of 4 is how much percent? 25%. 223, explain why this disorder, although it is sex-linked, does not affect males only. Now, we know it's caused by a dominant allele. It's caused, by a dominant allele, which is carried on the X chromosome. And we know both males and females have X chromosomes. 224. This genetic disorder is caused by a gene mutation in which the DNA triplet CAG is altered to TAG. Now, can you see the T replace the C? What type of mutation it is? Can you see it's a single nitrogenous base that's been replaced? So, of course, it's a point mutation. Describe how this type of mutation mentioned, which is a point mutation, will affect the structure of the protein it codes for. Of course, a different amino acid will be coded for, resulting in a different protein. Nice question. And I know you have not learned point mutations. You learned them quite briefly, but in the past we used to learn them in detail. 225. Give 2 reasons why this couple should undergo genetic counseling before having children. Now, they want to see if their children are going to have rickets, so that's why they must go for counseling to see what's the chances of them having a child with this disorder. The counselor will help them teach them to cope with having such a child and decide if they're going to make an informed decision and decide if they want to have the child or not. Right. 2.3. Ribonuclease is an enzyme made up of 127 amino acids. 231. What is the minimum number of DNA bases needed to code for amino acids of this enzyme? It's easy. There's 127 amino acids times 3 because there's always 3 nitrogenous bases, and your answer will be 381. Then 232. The sequence of DNA bases coding for 7 amino acids in the enzyme ribose nuclease is GTT, TAC, TAC, TCT, 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 TTA. Now, the number of each type of amino acid coded for by the sequence of DNA is shown in the table. Right. The first amino acid in this enzyme is gly, which is coded for by GTT, worked out in the DNA. Use the DNA base sequence and the table above to work out the sequence of the remaining amino acids in this part of the enzyme. This is a very nice question. This is why I chose this question paper. The types of questions they ask are very challenging and tricky. Now, can you see it tells you the first amino acid in this enzyme is gly, and it's coded for by GTT. So you know GTT is the first one and gly is the first one, so you don't have to write it. Then can you see there's 2 TACs? So which amino acid codes for 2 amino acids? So it'll be meth. Right. And you can see how it's written there. And then can you see TCT is coded for by 3. So what codes for that amino acid? Arg. And you can see how it's written here 3 times. And then the last one, which is not used, is ASIN, TTA1. Okay. This is a very nice question. Make a note of it, write it somewhere. It's a lovely question. Right. Study the diagrams below representing various phases of meiosis in our organism. So there you have diagram A, B, and C. 241. Which diagram A, B, or C represents meiosis 1? Meiosis 1. Can you see? It's diagram A. 242. Suggest why the chromosomes in diagram B will be genetically different from that of the parent cell at the beginning of meiosis. Now you know. Why is it different? What took place? Crossing over took place. And what takes place there? There's an exchange of genetic material, or you could have said random assortment of chromosomes occurred at the equator. Any one. It's only 1 mark. 243. How many chromosomes will each daughter cell have at the end of the cell division? 2. You can see that there in the diagram. 244. Give 2 reasons why this type of cell division is important. This is meiosis. Why is meiosis important? Meiosis increases genetic variation. Meiosis reduces the number of chromosomes by half. Meiosis results in the formation of gametes. And meiosis ensures that the chromosome number remains constant within the species. Right. 3.1. Professor Lee Berger and his colleagues studied the fossils they found in a cave at the cradle of humankind. The skeletons from the cave are ranked amongst the most complete finds to date. The adult female Australopithecus sediba was remarkably well preserved and some of the following characteristics were identified. Some characteristics of Australopithecus sediba: a small brain size, bipedal, small canines, projecting nose, and a small body size. Right, 3.1.1. Write down 3 characteristics from the list above that also apply to Homo species. So, projecting nose, small canines, and bipedal. 3.1.2. State 2 advantages of Australopithecus sediba being bipedal. Now, why did they become bipedal? They had a wider view to spot danger, they could carry their offspring or food or tools, and they had a larger surface area for thermoregulation. And 3.1.3. Professor Berger called Australopithecus sediba a transitional fossil. Using the information in the list above, explain why Australopithecus sediba is a species in transition. Now, can you see sediba has both characteristics of Homo and the Australopithecus species. So, a transitional fossil is intermediate between 2 groups and will have characteristics of the 2 groups. Right. 3.2. Study the table below which indicates some of the hominid fossils found in the world and answer the questions that follow. Now in your table you can see the different species: Australopithecus afarensis, Australopithecus africanus, Australopithecus sediba, Homo habilis, Homo erectus, Homo heidelbergensis, Homo neanderthalensis, and Homo sapiens. The regions they were found: Eastern Africa, Southern Africa, Sub-Saharan Africa, Africa, Europe, Asia, Europe and China, Europe, Western Asia, and worldwide. And there's the period of existence. 3.2.1. Explain why the information in the table supports the out of Africa hypothesis. Now, can you see that the oldest of the hominids, like the Australopithecans and the Homo habilis, are only found where? In Africa. Okay? While the younger fossils are found worldwide. Okay, what does that suggest? That humans originated in Africa. The oldest Homo erectus as well is found in Africa, while the younger ones are found in Europe and Asia, which suggests that Homo erectus migrated out of Africa. 3.2.2. If a fossil of Australopithecus afarensis dated 3.2 million years old is found in Asia, explain the implications for the out of Africa hypothesis. Oh, very nice question. This will mean the hypothesis will be rejected because it will imply that the origins of humans is not Africa but Asia. A very lovely question. Make a note of this. 3.2.3. Describe 2 genetic lines of evidence that support the out of Africa hypothesis. You could have said mutations in the mitochondrial DNA can be traced to the female ancestor in Africa, and mutations in the Y chromosome can be traced to a male ancestor in Africa. This was a very nice question. Right, 3.3. Study the analogous and homologous structure below, which can be used as evidence for evolution, and answer the questions that follow. 3.3.1. Identifies 2 structures in the diagram that are analogous. Now, analogous means they have different structures, but they have the same function. Now, can you see? It's the insect wing, it's the wing of the insect, and the phloem of the bat or the wing of the bat. Right, identifies 2 structures in this diagram that are homologous. Homologous means they have similar structure, but for different functions. So can you see? Phloem of the human and the wing of the bat. Right. What are homologous structures? They ask you 3.3.2. Describe how the homologous structures named in question 3.3.1 give evidence for evolution. Now, what does it prove? These organisms have the same structure, which originate from the same ancestor. You see, this proves the fact that they all have a common ancestor, these different organisms. Right. The diagram below shows 5 closely related species of finches found on the mainland and on 4 nearby islands. So there you have them. Island 1, 2, 3, 4, and then the mainland. Describe how the different species on the 4 islands evolved over many generations from the original ancestor onto the mainland. This is a very nice question. Question 3.4 is very nice. The finch, these are finches. There was a variation in the different beak sizes. The population was separated by a geographical barrier. Okay. Allopatric speciation took place. We are not in 2022 allowed to call it allopatric speciation, but in 2013 we are allowed to. Right. As the different islands had different environmental conditions, such as different food, different vegetation, each group on each island underwent natural selection independently. And what happened? They developed differently. They became genotypically and phenotypically different. The gene flow or reproduction between the different populations did not occur, resulting in different species forming. In bold, that's the compulsory marks, meaning you must have those 2 marks. If you don't have them, you don't get your full marks. Right. 4.1. A group of learners investigated the frequency of dominant and recessive traits in their schools. Their hypothesis was there will be more learners with dominant traits than learners with recessive traits. The traits below were investigated in 200 learners. So there you have the dominant trait, unattached earlobe, rolled tongue, bent little finger, and the recessive trait was attached earlobe, unrolled tongue, and straight little finger. The results below were obtained during the investigation. So here we have the different traits, the number of learners for dominant and recessive. Now, 4.1.1. Show the results for each trait above as bar graphs on the same set of axes. So, they want the results on the same bar graph. So, what must you have as your heading? The frequency of 3 different traits in the learner population, one axis, number of learners, then the traits, and then your key, earlobe, tongue, little finger, and then clear is dominant and shaded is recessive. You got a mark for the correct type of graph, the heading for the graph, the label for the x-axis, and the width of the bars had to be the same. The graph labels at the below the graph, the key meaning had to be correct. Correct labels for the y-axis and the scale, and all the bars had to be plotted correctly. 4.1.2. Explain why age need not be controlled in this investigation. These traits that we just discussed are inherited and not influenced by age. 4.1.3. State one way in which the learners ensured reliability. Now, look at that. They had 200 learners, so they had a very large sample size. 4.1.4. State any 2 planning steps that the learners had to consider before conducting the investigation. So what must they do before they conducted the investigation? Get permission from the principal or the authorities to conduct the investigation, decide on the appropriate time of day to conduct the investigation, decide on the sample size, decide on the sample selection, identify each trait, so they had to learn how to identify each trait and decide how to record results. 4.1.5. Will the learners accept or reject the hypothesis? That's easy. Rejected. Why? Because most learners displayed recessive traits compared to dominant traits. Right. In mosquitoes, there is a gene locus which has alleles involved in resistance to DDT, a well-known insecticide. The graph below shows the number of mosquitoes and their genotypes collected from 1965, when DDT was first used, until 1970, 2 years after the spraying of DDT stopped. So here in your graph, you have the number of mosquitoes, 1965 when spraying started, in years, 1968 when the spraying stopped. 4.2.1. How many alleles are involved in the trait for resistance in mosquitoes? You can see from this that it's only 2 alleles, big R and small r. 4.2.2. State the genotype for resistance to DDT. So that will obviously be small r, as you can see in the graph. 4.2.3. Which genotype appeared most frequently in the population in 1970? In 1970, can you see? Big R, small r. And 4.2.4. Which genotype's chances of survival was reduced after spraying DDT was stopped in 1968? That's easy. Big R, big R. Right, and the last question. Charles Darwin and Jean-Baptiste Lamarck have different ideas to explain evolution. Describe how each of them would have explained the evolution of the long necks of giraffes. Justify whose idea is more acceptable in science community today. Even though you don't get asked an essay, these essays can be taken and broken up into short questions. That's why I still do them in my videos. So let's look at Charles Darwin's explanation. As a result of genetic variation, some giraffes have long necks and others. Environmental change occurred when the leaves became scarce in short trees, so there was competition for resources that occurred, causing those giraffes with short necks to die and those giraffes with long necks to survive. This is called natural selection. The genes for long neck was passed on to the generations, and as a result, all the population of giraffes have long necks. maximum 8 marks. Jean-Baptiste Lamarck's explanation, all giraffes had short necks originally. When the leaves became scarce in short trees, in the lower parts of trees, the giraffes stretched their necks, right? And they used their necks more often to reach the taller trees. As a result, the neck became longer. The acquired characteristic was passed on to the offspring, and the next generation of giraffes had long necks. Now, an idea accepted in science community today, Charles Darwin's is accepted. There is evidence that genes that are inherited from parents, right, that we inherit our genes from our parents and that it's not acquired. It's not something that you just get. Well, grade 12s, that was a nice paper for me. It was one of the papers that has a lot of challenging questions and it makes you think. I hope you learned a lot. Thank you for watching this video. Join me and watch all my other videos as well.